Good morning. I would like to welcome all of you to the first session of the fourth scientific summit on tobacco harm reduction. During the following 1 hour, we will have the opportunity to discuss interesting research issues about toxicology, biomarkers, preclinical evaluation, and epidemiology. I think that if we are ready, we can start at once. Thank you very much.
Hello to everyone. My name is David Bovard, and the title of my presentation is Aerosol from a heated tobacco product has less effect on liver detoxifying enzymes than cigarette smoke. As you have seen from the title, the topic of my presentation is about the liver. This organ is involved in the production of albumin, bile, it stores vitamins, it also processes glucose and detoxifies xenobiotics. For this last function of the liver, the first step of detoxification is performed by a family of enzymes named cytochrome P450. This family is composed of 57 different enzymes, with each of them detoxifying specific substrates. I give you here two examples of these enzymes. So you have CYP3A4, which metabolizes caffeine, paracetamol, and you can also see CYP1A2, which metabolizes diazepam, amiodarone. You can also see that these two enzymes, they have a common substrate, which is clozapine. The activity of these enzymes can be modulated by many substances to which we are daily exposed. I give you here again two examples. You can see that CYP3A4 is inhibited by grapefruit, but CYP1A2 is activated by cigarette smoke. This modulation can be problematic when a person is exposed to several substances at the same time. On this slide, I give you an example. In non-smoker, so people who have a normal CYP1A2 activity and that are under clozapine medication, they will metabolize the parent compound, which is also the active compound, into non-reactive metabolites. In smokers, as we have seen, the activity of CYP1A2 is increased, which means that people under clozapine medication will need to intake more clozapine to have a similar effect as in non-smokers. If a smoker stops smoking, he will need to adjust the clozapine intake to match with the CYP1A2 decrease in activity. If it doesn't, there is a risk of over-medication with potentially harmful effects and even death. To prevent potential interaction with drugs, it is important to screen whether a product interacts with CYP enzymes. We, for example, question whether liver CYP enzymes are also affected in users of a tobacco heating system marketed under the brand name IQOS and referred to as THS for the rest of the presentation. So the aim of our study was to develop a physiological in vitro aerosol exposure system to detect the potential effects of aerosols on liver tissue viability and CYP activity. Our exposure system is composed of a 3D liver model. We used liver spheroids, which is the test organ, the organ on which we want to see a change in CYP activity. We used a 3D lung model, in this case bronchial tissues, which is the entry route for the aerosol. We used a multi-organs on a chip device, the Vitro fluid. I will come back on this device in the next slide. And a direct aerosol exposure system, the Cultex RFS. I will also explain a bit better this device in the next slide. The system we developed should answer the following questions. Can we mimic in an in vitro system the effect of cigarette smoke on liver tissues as observed in smokers? What are the effects of heated tobacco, THS, on CYP enzymes in the liver? As mentioned earlier, we used the Vitro fluid chip. The Vitro fluid is a multi-organ on a chip device, which is used to connect two different in vitro models by using a chip plate and a peristaltic pump. We use this device to co-culture lung and liver tissues. As I mentioned earlier as well, the Cultex RFS has been used to expose our 3D air-liquid interface tissues to cigarette smoke or to THS aerosol. As a first step in the development of our aerosol exposure system, we determined the maximal subtoxic dose of cigarette smoke and THS aerosol to which the lung tissues could be exposed. We proceeded as follows. We first exposed the bronchial tissues to a dose range of cigarette smoke, THS aerosol, or fresh air using the Coltex. We then placed the tissues in the Vitro fluid with the respective cell culture medium and incubated the tissues for 48 hours in the chip before we measured the ATP content. And we also measured the level of LDH in the cell culture medium. It's a marker of cytotoxicity. For all the data that I will be presenting on these slides, but also on the next slides, we always compared the results obtained after cigarette smoke or THS aerosol exposure with the data obtained in air-exposed samples. We observed that cigarette smoke was cytotoxic at doses above 16 puff, as it reduced the ATP content in the bronchial tissues and increased the secreted LDH in the medium. In comparison, the THS aerosol exposure did not affect tissues viability, even at doses equal to 56 puff. On the basis of the dose range, we decided to use up to 16 puff of cigarette smoke and 56 puff of THS aerosol. Using the maximal subtoxic doses of cigarette smoke and THS aerosol, we developed an exposure method to detect effects on CYP activity in the liver tissues. As you can see on the left image, we first moved the bronchial tissues into the Coltex RFS system and exposed the tissues to cigarette smoke, THS aerosol, or fresh air. We then moved the tissues to the Vitro fluid chip with the medium. At the same time, we also placed 30 liver spheroids in the second compartment of the chip and incubated the two tissues for 24 hours with medium flow. After the 24 hours, we exposed the bronchial tissues again using the Coltex RFS to THS aerosol or cigarette smoke before placing the tissues back in the Vitro fluid. After 48 hours of incubation, we measured CYP1A1, 1B1 activity in the bronchial tissues, CYP1A1, 1B1, CYP1A2, and CYP3A4 activities in the liver spheroids. And we also measured LDH in the medium. As you can see, and using this approach, we did not detect LDH in the medium, confirming the absence of cytotoxicity for the 3 doses of cigarette smoke and the 2 doses of THS aerosol tested. In the bronchial tissues, we observed a strong CYP1A1, 1B1 induction after the exposure to cigarette smoke. As CYP1A1, 1B1 has a high activity in the lung of smokers, it confirmed that our cigarette smoke exposure was successful. You also see that THS exposure did not affect CYP1A1, 1B1 activity. We then tested the activity of CYP1A1, 1B1, CYP1A2, and CYP3A4 in the liver tissues and found a dose-dependent increase in CYP1A1, 1B1 and CYP1A2 activities after exposure to cigarette smoke. While CYP1A1, 1B1 activity was increased only by 20% after cigarette smoke exposure, CYP1A2 was increased by up to 150% when compared to the activity in the air-exposed tissues. CYP3A4 activity was also induced by cigarette smoke, but the effect was weak and dose-independent. THS did not cause any significant effect to CYP1A1, 1B1, and CYP1A2. There was a slight decrease in CYP3A4 activity after exposure to the THS aerosol. As the method developed was resulting in robust data with similar in vitro effect as observed in smokers, we tested 3 other CYPs. We did not detect any effect of cigarette smoke exposure on CYP2B6, CYP2C9, and CYP2D6 activities. While CYP2B6 and CYP2C9 were not impacted by THS aerosol exposure, we detected a small decrease in CYP2D6 activity when compared to the air-exposed samples. Because we use bronchial tissues for the exposure to cigarette smoke or to the THS aerosol, we also evaluated CYP activity in those tissues 48 hours after the last exposure to cigarette smoke, THS aerosol, or fresh air. And we observed that after cigarette smoke exposure, in addition to CYP1A1 1B1 induction reported earlier, CYP1A2 was also induced by fourfold in the lung tissues, and we also observed a decrease in CYP2B6 and CYP3A4 activities when compared to the air-exposed tissues. No significant effect was observed in the lung tissues after exposure to THS aerosol. As a summary of our study, we have observed that using our physiological aerosol exposure system, we detected an induction of CYP1A1 1B1 and CYP1A2 in the lung tissues. And cigarette smoke induces CYP1A1 1B1, CYP1A2, and CYP3A4 in the liver tissues. These in vitro data match the in vivo data from the literature, although the CYP3A4 inhibition was only reported in rodents. THS aerosol, at doses 4 times higher than cigarette smoke, did not cause any cytotoxicity. It slightly decreased CYP2D6 and CYP3A4 activities in the liver and had no other effect on CYP activity. Because THS has less effects than cigarette smoke on CYP activity in lung and liver tissues, THS users are less likely to suffer from drug interaction than smokers. This is the end of my presentation, and I would like to thank all the people present on this slide for the help during the study, and you for your attention. Thank you.
In this study, we investigated the possible effects of reduced danger smoking products on the development of age-related macular degeneration. Age-related macular degeneration is a degenerative disease that usually occurs in people after the age of 60. The main target is the retinal pigmented epithelium, leading to compromise of the blood-retinal barrier, death of photoreceptors, and finally loss of vision. Statistically speaking, it is the third most common type of legal blindness, and by 2040 it is estimated that almost 300 million people will suffer from a form of the disease. Advanced AMD can be categorized into two different types. The first one is the wet AMD, which combines death of photoreceptors and neovascularization. The second type is the dry or atrophic AMD. This type is characterized by severe damage to retinal pigmented epithelium and inability of the tissue to repair it. There is a strong positive correlation between smoking and the occurrence of age-related macular degeneration. Epidemiological data connect the years of smoking with increased chances of developing AMD. In Australian people, there is a 1.6 times increased occurrence of the disease in people smoking for at least 20 years, with the number going up to almost 2.4 times for people smoking for at least 40 years. Same numbers have been reported from researchers in USA and Europe. In addition to clinical observation, a series of in vitro publications emphasize the negative effect of cigarettes on retinal pigmented epithelium cells. The main effect observed by most works is the reduced antioxidant capacity of cells after exposure to smoke. Until now, there are no data on how alternative sources of nicotine consumption can affect the development of AMD. In this study, we investigated the effect of tobacco heating products and electronic cigarettes on a series of markers related with AMD. For the tobacco heating products, we used the commercially available IQOS with the red selection sticks, and for the electronic cigarettes, we used a laboratory-made liquid with 18 mg of nicotine per ml. For all the experiments in this present study, we used the retinal pigmented epithelium cell line ARP19. The chosen method of exposure was by producing and administering smoke or vapor enriched media. For this purpose, the LM4E and LM1 machines by Bovard were used in order to recreate the proper smoking protocols. For conventional cigarettes and IQOS, the Health Canada Intense protocol was used, while for the electronic cigarettes, the CRM81. First, the nicotine concentration of the enriched media was determined by using UPLCMS. As you can see in the presenting table, all three enriched media had similar concentrations of nicotine, and from this point on they considered to have the same concentration. Different concentrations of enriched media were used in order to determine any possible changes of the viability of cells. Cigarette enriched media had the most profound effect on cell viability. At 5% concentration, it was able to reduce the viability to almost 50%. At this concentration, the other two products had no effect on viability. The first adverse effect by the ICOS-enriched media was observed at the 50% concentration, where the cigarette media was able to reduce the viability to almost 10%. On the contrary, the e-cigarette enriched media was not able to reduce the viability at even 100% concentration. After the initial viability assays, the 5% concentration was used on all the further experiments. As in viability assays, conventional cigarette had the most profound effect on the antioxidant capacity of ARP9T cells. On the left diagram, cigarette-enriched media reduced the amount of superoxide dismutase to 65%, while the other two media reduced it to 85%. The effect on glutathione level was even more severe, with cigarette reducing the available glutathione to 30%. Apart from that, only an electronic cigarette was able to reduce the glutathione levels, but to a smaller degree. No change in the antioxidant capacity was observed after administering ambient air-enriched media. The same thread followed the three apoptosis markers that were investigated. Only cigarette-enriched medium was able to increase the expression of caspase-3 by 2.5 times, while no other media had any effect on this marker. Similar effects were also observed at the levels of activated caspase-3, increasing the amount of the activate protein by 2 times. On the last apoptosis marker, the BAX protein, cigarette-enriched medium was able to increase its expression by 47% and electronic cigarette-enriched medium by 30%. At the same time, ICOS and ambient air media had no effect on BAX protein. The last category of biomarkers targeted cellular senescence. The levels of the first marker, P16, were elevated only by cigarette, while the tobacco heating product and the electronic cigarette had no effect on the expression of P16. Again, only conventional cigarette increased the expression of the second senescence marker, P21, while the other two products had no change of statistical importance. To sum up, this study confirms the published data on the effects of smoking on the development of age-related macular degeneration by examining its effect on a broad selection of markers. Due to the less harmful effects of tobacco heating product and the e-cigarette on viability, apoptosis, antioxidant capacity, and cellular senescence, we can conclude that people using them could have lower chance of developing age-related macular degeneration. Perhaps the most valuable conclusion from this study is that since the alternatives have less impact on AMD, could be used as a suggestion to smokers with newly diagnosed AMD in order to avoid further deterioration.
Hello, I am Teemu Kärkelä and I am presenting our work on the absence of solid particles generated from thermal processes in the aerosol from an electrically heated tobacco product. Heated tobacco products were recently developed as better alternatives to cigarettes for adult smokers. One such HTP is the electrically heated tobacco system from Philip Morris International, also known as Tobacco Heating System 2.2 or ICOS. The working principle of the EHTS is that the tobacco portion of the consumable, or tobacco stick, is heated instead of being burned, with temperatures not exceeding 320 degrees Celsius. And this results in a very important reduction of harmful and potentially harmful constituents. A previous study has demonstrated that cigarette smoke contains carbon-based nanoparticles in contrast to the EHTS aerosol, for which no combustion-related solid particles were observed. One possible hypothesis to explain the absence of combustion-related solid particles in the EHTS aerosol could be that the different filter elements of the EHTP remove these particles if they are at all generated. To assess this hypothesis, modified EHTPs without the filter elements were produced for the purpose of this study. And then the results were compared to the reference cigarette 3R4F. We had two approaches in use, and the first approach was based on the aerosol Diluso method. So the tobacco product was connected to the puffing machine, and the sample was diluted, and then particulate matter was collected on TEM grid. You can see the TEM grid here. So it's composed of a copper grid where there are carbon foils, and then there are holes in the carbon foil. Flow goes through the hole, and then particles are collected on the foil. In the second approach, we were using thermodynamic aerosol method. So we connected our tobacco product to the puffing machine, and then there was a dilution step, and after that there was a thermodenuder which removed volatile gaseous species, and the particulate matter was collected using impactor. And here you can see an example of an impaction stage. So we did several tests, and we were using two different test methods. And we were using EHTPs with or without filter elements, and the EHTP was heated or non-heated. And the results were compared with the 3R4F research cigarette. The puff number ranged from 1 puff to 12, and we were using Health Canada intense regime as test conditions. So here we have results. With EHTP without filter elements and no heating, there were basically no deposit on the grid surface. However, we observed some large fragments, but we think that these fragments are coming from the tobacco leaves when the airflow is stripping material from the tobacco. Then with EHTP without filter elements and with heating, we noticed that there is some deposit formation. So we had this kind of gray colored areas. And when we heated this grid up to 200 degrees Celsius, we noticed that this deposit area was completely vaporized from the grid surface. Then with EHTP with filter elements and with heating, we noticed that there is once again a similar kind of deposit layer formation. And when we heated up this grid up to 200 degrees Celsius, we noticed that the deposit layer was vaporized. Next, we were testing 3R4F reference cigarette, and we were of course burning it, and we had 10 puff per cigarette. And on the other hand, we were also testing the reference EHTP, which was also heated, and we had 11 as puff numbers. In both cases, we had quite thick deposit layer formation on the grid surface. And when we were heating these grids up to 200 degrees Celsius, we noticed that for the EHTP, the deposit layer was vaporized, and there were basically nothing on the grid. And for the 3R4F, also the deposit layer was vaporized, but this time we noticed that there were solid particles as a residue on the grid surface. So there was a clear difference between these products. For the cigarette, we had solid particles, and for the EHTP, there were no solid particles. The same results are also shown in this summary, and this is from using the method B. And here we have results for the supermicron particles which were collected using Impactor. So here we have results for the 3R4F research cigarette, and as you can see we have a lot of particles on the impaction stage. Then for the EHTP, with or without filter elements and with or without heating, there are basically no particles on the impaction plate surface. And for the submicron particles we have similar results. So for the 3R4F research cigarette we have a lot of particles, and for the EHTP with or without filter elements and with and without heating, we notice that there are no particles on the impaction plate surface. So our results indicate that unlike cigarette smoke, which consists of both liquid and solid particles produced during tobacco burning, the liquid-based EHTP aerosol contains no solid particles related to either tobacco heating or combustion, and combustion is not taking place in the EHTP. Thank you for your attention.
The title of my presentation is Combusted Cigarettes versus Heated Tobacco Products, Comparison from the point of view of particulate matter emission and soluble matter. As we all know, there are a number of health problems related to cigarette smoke, which have led to the promotion of measures for smoking cessation. For those who cannot quit smoking, the design of tobacco-related products with less or non-harmful effect, such as heated tobacco products or HTPs, is a valid alternative. Considering this, a question that needs to be answered, supported by independent studies, is the following. Are heated tobacco products less harmful than regular cigarettes? Thus, our research aims to compare combusted cigarettes with HTPs, and with such purpose, an experimental setup has been designed and optimized to simulate puffing and to collect in water any particulate matter or water-soluble compound. Such comparison, combusted cigarettes versus HTPs, is of great relevance, and in the last years, several studies have focused on the analysis of cigarettes and heats, paying special attention to any particulate matter generated. In the previously mentioned published studies, such particulate matter is usually collected from the aerosol phase, and in the present independent study, we propose to focus on the collection in water media and the characterization of any particulate matter and soluble compound. Note that this collection way resembles the interaction between the aerosols and water-rich media occurring in biological tissues. The present slide shows a scheme of the designed experimental setup, which has been optimized to fit the Health Canada intense puffing regime. Using this setup, 15 cigarettes have been smoked with puffing and compared with the use of 15 HTPs in terms of particle matter and soluble compounds collected in water, as aerosols bubble through it. See the water media in the gas washing flask. Detailed information about the design setup and the collection procedure appear in a recently published paper, whose reference is included in the slide. So, this setup has been used for doing experiments based on smoking 15 cigarettes or using 15 heats, which are the HTPs developed by Philip Morris products. In these experiments, the aerosol generated when smoking passes through the water contained in the gas washing flask. Those water samples have been collected, and the present slide shows pictures from both experiments, with cigarettes, top photograph, and with heats, bottom one. From them, the difference in color can be clearly observed. Taking into account that one of the main aims of this research is to analyze any particulate matter in the aerosol from cigarettes or heats, in some particular cases, the washing flask has been filled not with water but with isopropanol. Why to use an organic media? Because this way, if the particulate matter generated has an organic nature, as expected, we could have some additional information. These two highlighted pictures correspond to isopropanol media from cigarette experiments, top image, and heats experiments, bottom image, and it can be clearly observed again, even more, the difference between the color of the isopropanol when cigarettes are smoked in comparison with the use of heats. These pictures mainly show the color, but if the solutions are observed from close distance, in the case of samples from cigarette experiments, solid particles can be visually distinguished. Let's start with the results section, with data from laser diffraction characterization, a technique used to confirm or discard the presence of solid particles and to characterize the particle size distribution. Water samples from cigarettes or heats were analyzed. Particles were detected in all cigarette samples analyzed. An example of the distributions obtained is presented in the slide. In contrast, particles were not detected in heat samples. Only in some portions of heat samples, some low-intensity signal was observed, in the range of the detection limit of the equipment. Transmission electron microscopy has also been used as a technique to analyze any particulate matter in the water media. The left images, C1 and C2, correspond to samples from cigarettes, and the right images, H1 and H2, to samples from heats. For cigarettes, a lot of particulate matter is observed, in form of homogeneous spherical structures. The corresponding size distribution is shown as well. In contrast, this does not occur for heat samples. H1 is a representative image for heats. During transmission electron microscopy analysis, it is very difficult to find areas where any particulate matter can be observed in heat samples. Just after a thorough search, some solid particles can be found, such as in image H2. Determination of the total organic carbon content in water samples from the two types of experiments with cigarettes and heats has been done to have some information about the content of organic carbon and hence about the soluble matter. The data presented clearly shows that TOC values from experiments with cigarettes are much higher than from experiments with heats, which means much higher content of soluble compounds in cigarette samples. UVVs has been used as a technique to provide information of the presence and concentration of organic compounds in the liquid media, either water or isopropanol, from experiments with cigarettes or heats. In the case of cigarettes, the solutions are so concentrated that only after dilution they can be measured. That is why, in the two series of the large graph, those corresponding to cigarettes, the initials dF from dilution factor appear. This does not occur for heats, being this a first indication of the much higher soluble compounds concentration in cigarette samples. After recalculation, so that a direct comparison of the series can be done, the small right graph clearly shows that very low concentration of soluble compounds is present in heat samples. So, finally, we would like to highlight that cigarette smoke contains solid particles generated during combustion, as it has been experimentally observed. In contrast, the presence of particulate matter in aerosol from HTPs is negligible and not related to combustion. A much higher amount of compounds dissolved in water is present for cigarettes in comparison with heats, and all these observations together let us conclude that HTPs are less harmful than combusted cigarettes.
I'm Konstantinos Misiakaris. I am pharmacologist researcher at Iris Institute in Patras Science Park. At Iris Institutes, we are running several projects. One of them is the evaluation of the toxicity of tobacco products on several models that we have developed. Today I'm going to present you our work on Joule Products, and my presentation has the title In Vitro Evaluation of Joule Different Flavor Products on Human Alveolar Cell Lines. As the cigarette market was growing at a fast pace, hundreds of devices, models have emerged. One particular e-cigarette device that has gained significant popularity over the past years is known as Joule. Joule is a leading e-cig brand in the US, and in 2018 controlled more than 70% of the cig market in the cig market shares. Even though Joule only began to be marketed in 2015, in 2018 it was the most popular brand used among teens and young adults in the United States. It consists of pre-filled pouch, and it is easy to use, while it is a no-button push puffing device. Compared with other products in the market, the liquid composition of the Joule pouch contains nicotine salt and higher concentrations of nicotine. This study examined the effects of Joule on human alveolar cell lines. Human alveolar epithelium is primarily affected after smoking and vaping, and that's why we are testing the oxidative stress and inflammatory markers on human epithelial alveolar cell lines. The cell lines that we are testing are the adenocarcinomic alveolar basal epithelial A549 cells and NCI292 bronchial mucoepidemic carcinoma cells. For condensates preparations that administrated in cells, Bovard LM4A smoking machine was used. The CRM 81 Coresta protocol was followed while puffing with a rectangle puffing profile. A high concentrated solution was created, which was administered in multiple dilutions. You can see the LM4A device to the left. The advantages of the device include the ability to precisely adjust the puff volume, the control for leaks between the joints, as well as monitoring of exposure conditions such as temperature and humidity. To the right, you can see the protocol pathing details, such as the path volume and the duration of each path. The device was placed at an angle of 45 degrees, and the aerosol generated was trapped in a single impinger setup. The condensate generated, filtered, aliquoted, and stored at -20. Moving forward to the results, MTT assay was used for cell viability. This graph shows the percentage of viability in relation to control for alpine, menthol, golden tobacco, mango, rich tobacco, and mint flavors. Various concentrations were administered. Despite the high concentration of the solution, the expected decrease in viability was not observed in any flavor. Statistically significant differences were observed only in 100% group in relation to the air. More specifically, in the menthol group, which with 25 dilution, the decrease was recalled. The results in the second cell line, NCI 292, have not yet been completed for all flavors. That's why I present only the alpine. The alpine flavor is completed, and comparing the viability between the two series, we notice that we have the same picture. Reduction in relation to air is observed only at 100%. Malondialdehyde results from lipid peroxidation and is a marker for oxidative stress. The degree of lipid peroxidation can be estimated by the amount of malondialdehyde. According to the results in this graph, we can observe that no statistical changes are presented compared to control. The second marker of oxidative stress is glutathione, which is an antioxidant capable of preventing damage to important cellular components by reactive oxygen species. Reduction is translated as oxidative stress. According to the results, a statistically significant decrease observed only in flavor-rich tobacco compared to control. The next one is superoxide dismutase sode, which is an important antioxidant defense. In this graph, sode activity is presenting. According to the results, statistically significant decrease observed only one flavor, the mint group, compared to control. Before moving to the conclusions, I'm gonna talk about our next steps. We repeat all experiments on the second cell line. We examine inflammatory biomarkers, and we will reproduce all experimentation on Alice setup. Air liquid interface cell culture, ALI, is a method of cell culture by which the cells are grown with their basal surface in contact with media, and the top of the cellular layer is exposed to air, smoke, or vape. For this purpose, we use transwells for the cell culture, and in confluence status, they are placed in British American tobacco standard exposure chamber. The monolayer resembles the epithelium and is exposed directly to smoke or vape with LM4E machine. Finally, the conclusions. Toxicity by loss of availability is observed only in very high concentration and only in one flavor. Oxidative stress is not presented extensively as we expected. MDA, GSH, and so markers are affected by one different flavor and a different flavor each time. And more conclusions will be made by repeating the experiments in second cell line and early setup. Thank you very much.
The replica project was born from an idea of COIAR, the Center of Excellence for the Acceleration of Harm Reduction of the University of Catania. Replica is a 3-year project with excellent partners from different parts of the world. The no-smoke team of the University of Patras from Greece, the Sbarro Health Research Organization of the Temple University from the United States, the Sultan Qaboos University from Oman, the University of Kragujevac from Serbia, the Federal University of Kazan from Russia, and the University of Pajajaran from Indonesia. The first year of Replica involved only 5 partners. Italy, obviously, the University of Patras, the Temple University, Sultan Qaboos University, and the University of Kragujevac. Detailed information on the project is available on this link on the project website. All the centers were equipped with a smoke laboratory capable of working in ISO conditions with the same smoking and vaping machines, the same climatic chambers and incubators with exposure systems in order to expose the cells to smoke and vapor at the air-liquid interface. This is an example of the Italian lab in the University of Catania with the smoking and vaping machines on both sides of the controlled temperature incubator and an adjustable aspirator above to eliminate the waste fumes. For the first year of Replica, we have chosen to replicate 3 studies on the cytotoxic and inflammatory effect of smoking and vapor on human bronchial epithelial cells, the H292 cells. We have selected the studies because they were conducted by the same research group and have the same setting, but they are conducted with 3 different products. In the first paper, the P1, the cellular model used is studied in depth, and the differences between different regimes of exposure, ISO and ACE, to cigarette smoke are evaluated using the reference cigarette 3R4F of the University of Kentucky and comparing the cytotoxic and pro-inflammatory effects of wall smoke and vapor phase of smoke obtained eliminating TPM and nicotine using Cambridge filter pads. Here we are focusing on the cytotoxic effects of this product. In fact, in the paper 2, the same cytotoxic assessment were conducted using 2 different tobacco eating products. And in the third study, the same assessment are repeated by exposing the cells to the vapor from 2 different electronic cigarettes. We had some differences between the smoking machines and electronic devices used in the original studies and in our study. For example, the smoking machine used for the original studies permit to dilute the smoke and vapor with air, while our machine deliver only undiluted smoke and vapor to the cells. Moreover, for some products, we have only found the updated version on the market. As an example, the 1R6F instead of 3R4F, the IGOS 3 instead of the IGOS 2, and so on. Therefore, probably with a different performance in terms of nicotine release. So we firstly had to assess the dose response curve of the cells with our equipment and to define our IC50, the concentration of smoke that is required for 50% inhibition in vitro. Therefore, we measured the nicotine released in the culture media in the cell exposure chambers, determining the nicotine value released by the 1R6F cigarette to reach the IC50 with our cells, and we have exposed with all the non-combustion products to a number of puffs needed to reach the same amount of nicotine in the culture media of the exposure chamber. Oh, sorry. Only wall smoke exposure led to detectable quantity of nicotine in the culture media, confirming that Cambridge filter pad is capable of retaining most of the nicotine without making it arrive in the exposure chamber and therefore on the cells. With the wall smoke exposure, we observed an increase in nicotine concentration with increasing puff number and more with the Health Canada intense regimen than with the ISO regimen, as expected. The TPM release measured by weighting the Cambridge filter pads before and after exposure under Health Canada intense regimen was also significantly increased compared to the ISO regimen. Using nicotine dosimetry data from wall smoke exposures, cell viability of the biological responses was presented against exposed nicotine in the cell media for each puff number. Therefore, the IC50 for nicotine from wall smoke under the ISO regimen was 4.18 micrograms per milliliters and was roughly the amount of nicotine released by 10 puffs. Otherwise, under the Health Canada intense regimen, the IC50 was 9.7 micrograms per milliliters and was reached after about 5 puffs. Based on calculated nicotine IC50 dose by wall smoke under the Health Canada intense regimen, we chose to conduct the experiments with the ends by using the number of puffs able to produce the amount of nicotine closest to this value across all products. Thus, for all the comparison experiment between the cigarette and the ends, the 1R6F was smoked by the LM1 smoking machine with the Health Canada intense regimen for 5 puffs. The vaping entry button activated cigarette was vaped by LM4E vaping machine like all the other hands, following a modified Health Canada intense regimen with 1 second of pre-activation for 10 puffs, as per the original paper. The Vape Stick Max, a puff-activated cigarette-like product, was vaped following the Coresta reference method 81 regimen for 25 puffs. The IQOS 3 was vaped using the Health Canada intense regimen without blocking the filter vents for 7 puffs, and the Glow Hyper was vaped by the same conditions for 8 puffs. For each exposure run, we prepared 3 contours: an air contour with the cells exposed to the lab air filtered with a Cambridge filter pad, an incubator, and an air-liquid interface control prepared for each exposure by placing cells respectively with and without the inner culture media in the incubator. Substantial interlaboratory variability between the 5 laboratories was observed for all ISO wall smoke exposure conditions. Linear regression analysis of ISO vapor phase dose response curves showed good reproducibility between each laboratory except for one. Variability in the results for Health Canada intense wall smoke exposure was observed only for 2 and 4 puff in one lab. We evaluated the laboratory performance for each exposure condition and product variability. Interlaboratory variability is low across all laboratories and products except for 1R6F exposure performed by one lab. 1R6F exposure showed the greatest interlaboratory variability. Indeed, interlaboratory variability deviation were greatest for 1R6F compared to other products. There was an overall significant difference in natural red uptake cell viability among all products compared with the air control, driven mainly by the effect of 1R6F cigarettes. No reduction in cell viability was observed after exposure to tobacco-eating products or electronic cigarettes. Thus, in comparison between products, only 1R6F cigarette differed significantly from the other products. Our results support the reduced potential of electronic cigarettes and tobacco-eating products relative to conventional cigarette in an in vitro model of bronchial epithelial cells, even under more extreme exposure conditions, like undiluted aerosol and smoke, than those of replicated papers. For future studies, our findings suggest that aerosol dilution and normalization of data to better reflect cell functionality rather than viability will be important for achieving the most accurate results. Also, an important milestone was the establishment of collaboration within the scientists that will expand teamwork and progress knowledge in the field of reduced risk, non-combustible tobacco and nicotine products. Overall, this study confirmed that most of the harm to bronchial epithelial cells arose from volatile compounds in cigarette smoke rather than TPM or nicotine, and support the use of ENZYMES in harm reduction strategies for smokers. Thanks for your attention.
Good morning. My name is Shoaib, and I'm here to present on the behalf of Philip Morris International. Today, my topic of presentation is toxicological assessment of the aerosol generated by a novel pin-based heating system in a human bronchial epithelial cells and organotypic small airway epithelial culture. The novel pin-based heating system is an electrically heating tobacco system that heats the tobacco to provide pulmonary delivery of a nicotine-containing aerosol. As shown on the left, the pin-based heating system is very similar to the tobacco heating system, THS. The most important difference is that it uses an internal pin instead of a blade. The table on the right compares both systems. As you can see, PHS generates similar level of nicotine and aerosol delivery as THS. During the presentation, I will use PHS for pin-based heating system and THS for tobacco heating system. The aim of this study was to compare the biological impact of exposure to the aerosols generated by PHS and THS. The biological impact of this aerosol were also compared with that of cigarette smoke from 1R6F reference cigarettes. To this aim, we use monolayer of normal human bronchial epithelial cells to perform real-time cell analysis and high-content screening in what we call the 2D part of the study. Additionally, 3D organotypic culture of small airway epithelial cells were used to assess the impact on gene expression, histology, cilia repeating frequency, barrier integrity, cytotoxicity, and secreted factors. For the 2D part result, real-time cell analysis. In the real-time cell analysis, the greater the electrode impedance, the greater the number of cells on the bottom of the well, and hence greater cell viability. For cigarette smoke, we found a dose-dependent reduction in the cell viability with a half maximal dose concentration of 0.045. On the contrary, there were no significant changes for PHS or THS aerosols. For the high content screening, the central figure shows the result of the high content screening. In this figure, the bigger the area, the bigger the effect, meaning that a lower concentration is needed to elicit this effect. With regards to the cigarette smoke has a similar impact at 4 hours and 24 hours. And decreased mitochondrial health membrane potential was one of the strongest effects, suggesting that it may be one of the first adverse effects occurring upon the exposure. Those effects have probably contributed to the observed increase of oxidative stress. Not surprisingly, we also found cell stress indicated by the increased translocation of fusion stress kinase along with the sign of DNA damage. With regard to the THS, we found a reduced impact on all analyzed endpoints when compared to cigarette smoke. Nonetheless, as with cigarette smoke was observed, a decreased mitochondrial health and increased oxidative stress with ROS and glutathione. No evidence of genotoxicity or hepatotoxicity was found. For PHS aerosol in general, the activated endpoint and corresponding minimal effectiveness concentration in PHS matched those of THS. For the histology part, the tissue were exposed, and after 48 hours of post-exposure histology, tissue was stained with 3 different types of stain: hematoxylin, iocine, and Alcian blue. The figure on the right shows a representative picture of 3 different cultures for each treatment. Culture exposed to THS or PHS aerosols had a morphology similar to the culture that were exposed to air for all puff tested. Culture exposed to cigarette smoke for more than 2 puffs had pronounced tissue damage and thinner epithelial culture. Cytotoxicity and barrier integrity was also measured. To assess cytotoxicity, we measured the activity of adenylate kinase in the basolateral media. The more kinase activity observed, the more leakage, and therefore the more cellular damage. Exposure to THS or PHS aerosol did not impact cytotoxicity. For the barrier integrated tear measurement was performed, epithelial barrier integrity was assessed, measuring the transepithelial electrical resistance or tear at 48 hours after the exposure. In contrast to cigarette smoke, tear values in the presence of PHS and THS aerosol were comparable to corresponding air-exposed culture. Ciliary beating frequency. Regarding ciliary beating, the upper graph shows the ciliary beating active area while the lower graph shows ciliary beating frequency. All culture had a similar baseline beating frequency and the active area. After the exposure 48 hours in general, cigarette smoke led to a reduction in the active area and the beating frequency. THS aerosol and PHS aerosol presented similar values for active area and the frequency. Those values were in general higher than in culture exposed to cigarette smoke. Concentration of secreted inflammatory mediator were assessed in basolateral media of the culture at 48 hours of exposure. As you can see in the figure, the concentration of several factors IL-1 alpha, IL-8 and MMP-1 increased with the puff number of the cigarette smoke. Exposure to the THS or PHS aerosols did not result in a dose-dependent alternation of any of the assessed inflammatory mediators. Gene expression, a causal network enrichment was conducted based on the gene expression profile of culture exposed to cigarette smoke, THS, and PHS aerosols relatively to air for 4 hours and 48 hours post-exposure timing. The network model was cell-fate, cell proliferation, cell stress, and inflammatory process network. The graph on the left shows the biological impact factor, BIF, which reflects overall impact on all network models. The highest BIF was observed with cigarette smoke exposure and defined as 100%, and this was the reference. In general, exposure to PHS or THS aerosol for any puff number resulted in lower BIF values and MPS scores than exposure to 1R6F cigarette smoke. Relative change comparison impact of THS versus PHS. The overreaching objective of this study was to determine whether the reduced impact of PHS aerosol was comparable and not worse that the reduced impact of THS aerosol both relative to cigarette smoke. This was considered fulfilled if the reduction of impact for PHS was at least 80% the reduction of the impact for THS. Result for cytotoxicity, impact of 8 and 10 puffs of cigarette smoke on cytotoxicity was expressed as 100%. The main reduction in cytotoxicity of PHS aerosol versus cigarette smoke were more than 80% of the mean reduction in cytotoxicity of THS. The relative change comparison impact of THS aerosol versus PHS aerosol for the tear and also for ciliary beating frequency and also for different secreted mediator were also measured. Value of 10 puffs of cigarette smoke were expressed as 1. The tear value associated with 10 puffs of PHS and THS aerosols were computed relative to this cigarette smoke reference group. Again, the impact of PHS was comparable to the impact of THS for same puff number. Similar results were obtained for ciliary beating values and secreted mediator for THS and PHS aerosols. The biggest difference was apparent for CXCL10, IP10, a pro-inflammatory cytokine triggered by IFN, which will increase around 70% relative to cigarette smoke for PHS and around 90% relative to cigarette smoke for THS. This was not considered clinically relevant. Or the conclusion for this study is like PHS and THS aerosol induced comparable cytotoxic and phenotypic effects, which are markedly lower than the effect induced by the cigarette smoke, 1R6F, in human bronchial epithelial cells. PHS and THS aerosol up to 50 puffs trigger fewer biological effect than cigarette smoke, 1R6F, up to 10 puffs only in human 3D organotypic small airway epithelial cultures. Relative change comparison allow us to conclude that the impact of PHS aerosol on organotypic small airway epithelial culture is comparable to and not worse than the impact of THS aerosol. Thank you very much for listening to this presentation.
So, hello to everybody. I'm really happy being together with all of you. Hello to all the friends and collaborators. Hello. And I would like to add that this kind of virtual meetings are not so, interesting and lively as the live meetings, but we have to be adapted. Anyway, I think that we have two questions from the audience. The first question is coming from Dr. Karahalil, and he's asking, how do you explain ICOS viability being lower than e-cig in zero media? I think, if I remember well, that this is for George Karanasios. George?
Yes.
I repeat it. If you, I think you can see the question in the chat.
No, can you please repeat it?
All right. Okay. How do you explain the fact that when you are administering ICOS, the viability is lower comparing to e-cig in zero medium media?
Well, the ICOS is a product that combines pressed nicotine tobacco leaves, so there has a more complex mixture of substances. Even in the case that it's of course better than the conventional cigarette, but still there are plenty of substances that have harmful effects to cells. Whether they administered e-cigarette had only propylene glycol, vegetable glycerin, and nicotine.
Okay. Yes, I think that the sticks of ICOS or any other heating product is the sticks are also having paper and are having the tobacco and not the e-liquids. So it is expected that this will happen. Thank you, George. There is another question from our good friend Solomon Ratamane from South Africa. We have discussed during the previous summits. And the question is, what were the problems with Joule products in the USA? Since Konstantinos Mesiakaris presented some, I think, results from Joule, although this is not connected with the presentation, I would like to give it to him, to offer to him this question. So,
yeah, I'm not sure
have
if
you
I can
heard?
answer that. Yeah. Of course, we are testing the toxicity of these products in order that the policymakers I can make some conclusions in the future. Maybe you could say more things about the Joule, Mr. Blas.
If I can understand, we are speaking about the recent problems that all the e-cigarette products are facing in the States. It has to do with the flavors and all this, but I think it is not an issue we have to discuss during this session. We have to wait for the FDA decisions. Is anybody else who would like to add something more for the Massimo?
I think it was a very interesting session, and I liked very much all the works presented. I will launch an idea here with other colleagues that, as you know, Konstantinos, with you and other partners, we are working on the replication of different papers as the presentation that I had before. And I think that it would be nice if many of these researchers could join for the future a similar experience with us or between them. It will be a way to produce strong results replicated in different labs. So I would thank you in your team for your amazing work with us and with your experiment, your proper experiment. And I'm really happy to see this activity in the field, and thanks for this summit. That is a very amazing summit.
Thank you, Massimo. The idea is interesting. However, between the presenters are industrial presentations, but however, there are some labs which are having interesting results. So the labs and the scientists can directly contact Massimo Caruso or myself in order to think, to collaborate in any future projects, either under COEHAR or under an Horizon project or whatever else. Because the concept is to make links between the labs.
I think collaboration is the key for our future. In many different forms. It's not important the logo, the principal logo, the K logo. It's important the collaboration between scientists.
So if there is no other question or any other comment, I would like to thank all of you for your participation. All the presentations will be on the website of the meeting, so everybody can visit again and again and can contact the corresponding authors or the researchers for any question. Thank you very much.
There is another question.
Yeah.
No, there is in the chat.
Ah, okay.
Not bad.
Have you done any research about second-hand smoke and third-hand smoke from tobacco, alternative tobacco?
Alternative tobacco products.
I think that second-hand smoke and smoke is not easily handled if we speak about research in toxicology and biomarkers. I expect the same question in the following sessions. I expect the same question in the following sessions, for which we will speak about clinical, maybe, research and clinical results. Okay. So thank you very much.
Thank you.
Thank you.
Enjoy.
Thank you very much.